Hello everyone, and welcome to our lecture on bacterial diseases. One thing that you will notice is that I don't have lesson objectives for the next couple of lectures, including this one, because really your objective is to learn everything that you possibly can about the diseases that we're covering. So everything that's in the lectures, everything in the book, those are the things that you want to know. You will, of course, have a study guide that will help you with all of this, but really the goal is to just absorb as much information as possible about these different diseases. Obviously, we can't cover every possible disease that is out there, so what I have done for the different organisms that we study, things like bacteria, viruses, protozoa, helminths, etc., is I've picked out some of the most significant ones. Like I said, there are plenty more out there. We can't cover every single one of them, but I try to focus on the most significant pathogens to humans so that you guys can get a good understanding of what the most common types of microbial diseases are. We are going to start our discussion about bacterial diseases with the genus called Staphylococci. Staphylococci is a fairly large genus, meaning there are several different species within this genus. All of them, though, are gram-positive cocci that form irregular clusters. You guys have seen those in lab under the microscope when you're doing things like gram staining, etc. And we find species of Staphylococci primarily associated with the skin and mucous membranes of animals. And most of the time, the animals that we find them on are mammals and humans, which are a type of mammal. But we can find them occasionally associated with other types of animals as well. Most of the time, though, when we think about Staphylococci, we're thinking about the ones that are associated with humans and other mammals. We can occasionally find them free living in the environment as well. Most of the time, Staphylococci are fairly adaptable to different environmental conditions. But for the most part, we find them associated with mammals and humans, or including humans. There are several species within this genera, but there are a few that are the most significant to humans at least. The most significant species of Staphylococci to humans are Staph aureus, Staph epidermidis, Staph capitis, Staph hominis, and Staph saprophyticus. And out of this list, there are three that are the most important, and those would be Staph aureus, if I can get my highlighter to work, there we go, Staph epidermidis, and Staph saprophyticus. So out of this list of the 5 most important species of staph to humans, these are the top 3. Overall, staph infections cause about 100,000 deaths in the US per year. And that is one of the reasons that we consider them to be one of the major pathogens. That's a fairly high death number overall when we're talking about living in the era of modern antibiotics and modern medical treatment. So this type of pathogen really is one of the most important ones for all of us to know about, whether we're in healthcare professions or not, so that we can help prevent those types of infections, know what they look like when we do get them, and make sure they get treated appropriately. Now, out of that list that I gave you of the 5 most significant staph pathogens, and the 3, when we narrowed it down to those 3 most significant staph pathogens, out of all of those, the most significant Staphylococcus pathogen is called Staphylococcus aureus. So we are going to focus our discussion from here on out when we're talking about staph on this particular species. That last slide, we were just talking about Staphylococci in general. And then on this slide and the next couple of slides, we are just going to be talking about Staphylococcus aureus because this is the most significant pathogen out of that big group of Staphylococci. This organism is found primarily associated with human skin and mucous membranes. We can occasionally find it on other animals, but almost always when we find Staph aureus, it's indicative that a human has been around. And the thing that makes Staph aureus so bad and so dangerous is that it has a lot of different virulence factors. Virulence factors are characteristics of pathogens, characteristics of bacteria or viruses or fungi, etc., that allow the organism to cause disease. So the term virulence factors in general is just kind of a general term, and it refers to any trait or characteristic that an organism has that allows it to cause disease in a human, an animal, a plant, etc. And for staph, it has a lot of different virulence factors. One of the things that is significant about staph in terms of virulence factors is that it is very metabolically diverse. And by that, what I mean is that it's able to tolerate a lot of different types of environments because it has the ability to use lots of different things for electron donors, basically as energy sources, as electron acceptors for making ATP. It can handle lots of different ranges in terms of salinity and pH. So basically, it is an organism that is very good at growing in, or at least tolerating, a lot of different environments. So it can tolerate at least a little bit of salt, a little bit of acid. It can tolerate not having oxygen, so it can be a facultative anaerobe. It can also tolerate oxygen, so it can use oxygen if it wants to or needs to. And so basically it just is really good at growing lots of different places. It's not quite a halophile and it's not quite an acidophile, but it's at least halotolerant and at least acid tolerant, basically. It also usually has a fairly decent range of temperatures that it can live at, or at least survive at. It may not grow very well in very cold or very warm environments, but it can survive at them, and then when the environment gets a little bit better for it, it can start to grow again. So it just has this incredible ability to grow almost everywhere, really. It also has a lot of different enzymes and exotoxins. In terms of enzymes that it produces coagulase, which causes blood to clot. It can also produce staphylococcus, which actually does the opposite of coagulase and dissolve clots. And it produces something called penicillinase, which allows it to break down penicillin. So this organism is able to make our blood clot. It's also able to dissolve any clots that we form in our bodies. Sometimes we want to form clots in our bodies to help stop bleeding or keep an organism located in a certain area. But if we form one of those clots and the bacteria wants to go around it, it just releases this enzyme and starts dissolving those clots. And then it's also highly resistant to many types of antibiotics because of this enzyme called penicillinase that breaks down penicillin and most of the other drugs that are related to penicillin. It also produces several different exotoxins. So some of those important exotoxins are leukocidin, which kills white blood cells. My dog is snoring, sorry about that. It will also produce exfoliative toxin, which causes layers of skin to separate from each other. It produces toxic shock toxin, which is a toxin with kind of wide-reaching effects in the body that can cause everything from extremely high fevers to liver damage to kidney damage to just kind of general organ failure. So that one's a pretty nasty one. And then it will also produce alpha toxin, which destroys red blood cells. So this organism is just very well adapted to be a pathogen. It can grow all kinds of areas. It can cause our body to form clots. It can dissolve clots that our body forms. It is resistant to most antibiotics or many antibiotics. It will kill white blood cells. It will kill red blood cells. It will separate layers of skin. It will produce a toxin that causes just general organ failure for lots of different reasons. And this isn't even the full list of what it can do. These are just the highlights of this organism. So it is, like I said, this is the most significant pathogen out of this group of Staphylococci. In large part because of the organism's metabolic diversity and all the different types of enzymes and toxins it can produce, it's very good at infecting us and causing lots of different types of infections, because basically it can grow pretty much anywhere in our body. So some bacteria can only grow in our skin, or only grow in our lungs, or only grow in our stomach, etc., or our intestines or whatever, and they're kind of isolated to there, so they can't spread too far. Staph is not like that though. Staph will grow anywhere in your body that it can get a hold of, basically. So when we look at staph infections, we kind of group them into three main categories. The first one being cutaneous infections. These are skin infections. Usually they're fairly localized infections, meaning they haven't spread too far. They are on the skin, haven't made it into the deeper tissues, haven't made it into the blood yet. And if we're going to survive a staph infection, this is the best type of infection for us to have. A systemic infection occurs when the organism spreads throughout the body, typically through the blood. It doesn't have to be the blood, it could spread in different ways, but oftentimes staph will get into the blood, start to spread through the blood, and then it will start to infect various different organs, whether that is our liver or our kidneys, our digestive tract. It can cause damage to the lymph system, cause damage to all kinds of different areas in the body. And once that happens, it can be very difficult to come back from that type of infection. The third category of infection that we have in terms of Staph aureus is what we call a toxigenic infection. And this is an interesting one because with a toxigenic infection, the organism itself stays local to one part of the body, whether that is just on the skin, or maybe it could even be an internal infection. But the bacteria doesn't spread, it stays in one place. However, it will produce a bunch of toxins that it starts to spread throughout the body. So even though the bacteria stays local to one area, the toxins will start to spread throughout the body and start to oftentimes cause cell and tissue death. Another interesting thing about toxigenic infections is that we can get them from food, at least in this case for Staph aureus. So when we have a cutaneous infection, we have a systemic infection, we definitely have the bacteria in our body causing damage. However, with a toxigenic infection, it is possible that the bacteria is in our body releasing toxins and they're causing damage. But it is also possible that we never actually came in contact with the bacteria itself. So this is a special scenario. This is not something that happens every day, at least hopefully not to most of us. But there are situations where staph can infect food. And then when the food is cooked or prepared somehow, the staph itself, the bacteria, will die. However, in that time that the staph was growing on the food, it was able to release toxins. And the toxins don't get destroyed in the cooking or the preparation process. When we ingest that food, the toxins from the staph are still there. The bacteria is dead, but the toxins are there. And then the toxins make it to our GI tract and cause usually about 24 hours or so of vomiting and diarrhea until the toxin is cleared out as a result of the vomiting and diarrhea. So if you're going to get sick because of staph, that type is probably the best kind, although I would advise trying not to get sick with staph at all, if at all possible. So in terms of how this organism is passed on or how it is we become infected with this organism, most of these infections are actually what we call endogenous infections. And what that means is that a bacteria that has lived with us, just as part of the normal bacteria that live with us, gets into a place in our body where it shouldn't be and starts to cause an infection. So not all of us carry Staph aureus, but there are many people who do, and you may have done a test in lab to check your nose and see if you were carrying it. So some people do, some people don't. It can be a temporary thing. Some people may carry it for a while and then it goes away. But people who are carrying it may end up having a situation where that bacteria gets into a place it shouldn't be, and our immune system is not able to handle that bacteria for whatever reason. We then develop an infection, and hopefully it just stays somewhere local, but it can start to spread and become a systemic infection as well. So it is fairly common. In fact, sometimes one of the most common ways for someone to get a Staph aureus infection is from their own normal bacteria, and this is called an endogenous infection. Now, the next most common way probably to get a staph infection is skin to skin. So if one person is carrying it, we'll say person A is carrying it and touches person B, some of that bacteria from person A may end up on person B. And if that bacteria gets into person B's body, into a place where it shouldn't be, then person B will develop a staph infection or may develop a staph infection. And then finally, through food. So like I was just talking about with toxigenic infections or toxigenic situations, we can have a situation where there was staph bacteria on the food, and while it was growing there, it had a chance to produce some toxins. Now, by the time we consume the food, the bacteria is long gone. Either it's just gone or it's dead as a part of the cooking or preparation process, but those toxins don't go away. So while the bacteria was growing on the food, the staph was producing those toxins, preparing the food, ended up killing it, but the toxins are still there. We ingest the toxins and then get sick for around 24 hours. Could be a little bit more, could be a little bit less. In terms of prevention for staph infections, one of the big things is hand washing. So hand washing before you're going to prepare food, of course, hand washing before you're going to interact with patients, hand washing just on a fairly regular basis to make sure that you are managing your own normal flora, etc. Another significant thing with staph infections is if you do have a patient who does have a staph infection, it is very important to properly dispose of contaminated materials. Make sure you're using disinfectants and antiseptics properly, because this is a fairly easy one to transmit from person to person, especially if you have someone that has an infection. And then limiting exposure by carriers is important too. My understanding is that this does not happen in Las Vegas, but in many hospitals, people will be tested for Staph aureus on kind of a regular basis. And if they are carrying it at some point, they may be moved to a different set of patients or a different floor or something to make sure they're not around the most vulnerable patients. So that may not happen in Vegas very often or at all from what my former students have told me, but there are places that do regular testing of people, of healthcare providers, to make sure that they are not carrying Staph aureus. And if they are, it's not like a crisis or anything, they just move to a different set of patients or a different floor. So maybe they're not working with burn patients or someone else who is extremely vulnerable to this type of infection. The next type of organism that we will talk about are the Streptococci. So this is another fairly big genus, meaning there are a lot of individual species in there. And there are 3 main ones that we'll talk about at least a little bit on this slide, and then one main one that we'll talk about on the next couple of slides because it's the most significant. But before we get to that, the Streptococci are gram-positive cocci as well. They are formed in chains. You guys probably saw those in lab as well, most likely when you were doing your gram stains and other types of things. In terms of distribution, where we find them primarily, again, on the skin and mucous membranes of animals. And again, we're talking mostly about humans and other types of mammals, although we can find them in other types of animals as well. We can also find strep free living in the environment, although also, again, most of the time when we find Streptococci, they are associated with some type of animal, especially a mammal or a human. There are 3 major species that are important to humans. The first one is Strep pyogenes, which causes strep throat and impetigo and can cause sometimes fatal infections. The next one is Strep agalactiae, which is also just more commonly called group B strep. This one is significant, but usually only very significant when we're talking about pregnant women about to give birth. So I'll come back to that in a second. And then our third one is called strep pneumoniae. This one causes pneumonia. Now, pneumonia is just a general term for a certain type of infection of the lung. So there are multiple different types of organisms that can cause pneumonia. Bacteria can cause pneumonia, viruses can cause pneumonia, fungi can cause pneumonia. Technically, you could have chemicals that can lead to pneumonia as well. But this particular bacteria is one of the most common causes of certain types of pneumonia. On our next couple of slides, we are going to focus mostly on Strep pyogenes because it is the most significant pathogen out of this genus. So Strep pyogenes is the one that causes strep throat, also causes impetigo, and can occasionally cause more systemic lethal infections. Not super common, but can happen. Strep agalactiae, this is the one I said I would come back to. This is most commonly called group B strep. And group B strep is, for the most part, fairly harmless unless you are a newborn baby. So Strep agalactiae is carried by many humans, kind of intermittently. Some people carry it all the time. Some people carry it periodically. Some people carry it every once in a while. Some people never seem to carry it. But basically, the time that we worry about this particular pathogen is if a woman is carrying it in her 3rd trimester of pregnancy, and there is a chance that the baby will be exposed during labor and delivery. So this type of strep can be found in various places in the body, but one of the places we tend to find it is in the vaginal canal. And if a woman has this bacteria in the vaginal canal before she gives birth, there is a chance that the baby could come in contact with this bacteria during a vaginal delivery. The bacteria will basically get into the nose, the mouth, potentially the eyes of the baby, and make it to the brain and cause meningitis and potentially death. So if we find a woman who is carrying it during her 3rd trimester of pregnancy, typically what will happen is penicillin will be given during labor and delivery, which will prevent the baby from getting sick from this bacteria if it comes in contact with it. And then the last one, like I said, is strep pneumoniae. This is a very common cause of bacterial pneumonia. It is a disease that we can vaccinate against, which is great. And most of the time, people who are not immunocompromised don't get this type of pneumonia, but it is possible, especially in very young children, in the elderly, or immunocompromised people. And so it is one that we can vaccinate against, especially for vulnerable populations who may end up developing this infection. So like I mentioned on the last slide, we are going to spend most of our time when we're talking about strep species, talking about this particular one, Strep pyogenes. In terms of its particular distribution, we find it primarily in the nose, in the throat, and on the skin of humans. We can find it in other animals too, but most of the time we're talking about finding it on humans. And at any given point in time, in any given population, around 5 to 15% of the people in that population will be carriers. So this is one that you may be carrying sometimes, you may not be carrying it other times. It kind of just depends on what you've come in contact with in the world recently. But in general, in any human population, whether you're talking about in Las Vegas or somewhere in Siberia or somewhere in China or somewhere in Africa or just anywhere in the world, around 5 to 15% of the people in that population, wherever they are, whatever population it is, will be carrying that at any given time. In terms of virulence factors, it does have some pretty significant ones. It has a very sticky capsule that helps it, one, adhere to the body, and two, resist things like phagocytosis. So our body has a difficult time destroying this particular bacteria because of that capsule. It also has some fimbriae that are, in addition to the capsule, very good at helping the bacteria stick to our body in various places. And it has a couple of different surface antigens. There are what we call the C carbohydrates, the M protein, and the lipoteichoic acid. And although each of them has a slightly different function, when we look at them as a whole, essentially what they are doing is they help make the bacteria stick better, again, kind of in addition to the fimbriae and the capsule, help it resist phagocytosis, and just help it survive that much better in our body. It helps it stick there, helps it really lodge itself in, and just makes it a better pathogen. Now, in addition to those factors that really are working for the bacteria in terms of helping it stick to a certain place, it also produces some pretty significant enzymes and toxins that will cause damage in our body. One of the enzymes it produces is a type of protease. Now, protease is a general type of term that just means an enzyme that will break apart proteins. And this particular protease focuses on the complement proteins. So if you think back to Unit 3 and you remember learning, hopefully, about the complement cascade and how the complement cascade works to create this big membrane attack complex that will insert itself into a bacteria and basically act like a pin puncturing a balloon. Well, this bacteria has the ability to break apart that membrane attack complex so that our immune system it will create it, but it will get broken apart and it's no good at that point. It also produces something called streptokinase, which is an enzyme that digests clots. So again, kind of like with staph, oftentimes we want to form clots in our body because we're bleeding or we're trying to isolate the pathogen and keep it in just one location. But this type of bacteria will just produce this streptokinase, this enzyme, that will dissolve the clots and allow it to move freely throughout the body. In addition to the enzymes, it will produce a couple of significant toxins. One is that it produces streptolysins. These are toxins that will break apart red blood cells. It causes red blood cells to lyse or break apart. So you can think about that. That's why it says lysin. It means it breaks things apart. And then some of them, although not all strains of this particular organism will produce this, but some of them will produce a toxin called pyrogenic or erythrogenic toxin. This one actually comes from a bacteriophage, and it will cause a big rash to form all over the body. So this type of toxin is only produced if the bacteria has previously been infected with a bacteriophage. If it hasn't, then it can't produce this toxin. But if this bacteria has been infected with a bacteriophage, it gains this new characteristic or this new trait, and it can produce this toxin, which causes this massive rash to form on the body and typically helps increase the fever that we have as a result of this infection as well. Now, in terms of the specific infections this type of strep tends to cause, most of them are divided into kind of two categories, either the skin or local infections or the systemic infections. For skin and local infections, one of the most common ones is called impetigo. That is the one here at the bottom that you're seeing around this child's mouth. It's very common in younger children. Basically what happens is they might get a small cut or a small abrasion near their mouth somewhere through various activities or for various reasons, and then the strep that's on their skin infects that cut. They get this kind of localized infection that tends to produce this kind of yellow golden crust around their mouth, sometimes around their nose. And typically it's fairly easy to clear up with topical antibiotics or, if necessary, oral antibiotics as well. Now, the other, probably most common infection, or at least most famous infection that strep causes, is what we call streptococcal pharyngitis, or strep throat. So with strep throat, basically what happens is strep infects the throat, obviously. Typically in the tonsil region, you can get some kind of satellite infections in some other regions in the mouth, but most of the time we're talking about an infection of the tonsils. And what happens there is the strep gets inside what we call the little crypts, or the little holes that the tonsils have, and kind of overgrows, and oftentimes leads to things like fever, body aches. Sometimes you will see pus forming in the back of the mouth near the tonsil area. Sometimes you'll see little red spots that form in the back of the mouth, either kind of on the roof, on the hard palate, or a little bit even farther back on the soft palate. And fortunately, this type of infection is still very responsive to antibiotics. Penicillin tends to work very well still, or its derivatives. Oftentimes, something like amoxicillin or something else could be given for this type of infection. But most of the time, as long as someone has access to antibiotics, this infection does not become life-threatening, and it is relatively easy to clear up. However, there are times where this bacteria will start to spread throughout the body. And if something like impetigo or strep throat is not cleared up, then you can get a disease called scarlet fever. And really what's going on with scarlet fever is just that the bacteria has started to spread throughout the body. Most of the time, by that point, it has been infected by a bacteriophage, like we talked about on that previous slide. And as a result, as the bacteria spreads, it starts to lead to kind of a bright red rash that is disseminated throughout the body. Most of the time you find it primarily on the trunk, kind of the chest and the abdomen, but it can spread to other regions as well. And this is kind of a serious situation because the toxins that the bacteria is producing as it starts to spread throughout the body, or even just if it's kind of a local infection but the toxin production gets out of control, start to do damage to things like the cardiovascular system. They can damage the kidneys, and they can just lead to long-term effects in the body. So while strep is something that we are able to treat fairly well, and most of the time people don't get serious infections from their localized infections, it can become very dangerous if it is not treated properly. And then typically the worst case scenario with strep pyogenes is something called streptococcal toxic shock syndrome. This is where typically, not always, but typically one of these infections have gotten out of control. The bacteria has started to spread throughout the body, and they start to grow in the blood and the internal organs, release a bunch of toxins, and will just lead to organ failure and organ shutdown. And once that starts to happen, it's very, very difficult to kind of restore someone to health from that situation because the toxins have done so much damage to various organ systems. In terms of transmission of this bacteria, it is typically spread through what we call direct contact. So again, something like skin to skin or oral contact, where one person is directly touching another person and spreading the bacteria as a result. It's also spread by droplets sometimes when someone coughs it can be spread throughout the air that way. I was going to say when someone sneezes as well, but that would be a rare situation. Could it happen? Yes, but it would be a very rare situation to spread to spread strep through sneezing. So primarily through things like coughing. And then fomites. Fomites are non-living objects in the environment that are capable of kind of harboring microbes and transmitting them. So for example, something like a pencil or a doorknob or something else in the environment, a toy maybe, could harbor strep and then pass it from one person to another. In terms of prevention, one of the big things is just preventing droplet spread, making sure people are covering when they're coughing, washing their hands to help limit the amount that might be transmitted from person to person that way. And then in certain situations, it may be important to limit contact with carriers. There are some people that carry this bacteria without any obvious illness or any obvious effects in their body. And so this is another one that, even though we might not test for it in like Vegas hospitals or Vegas clinics, there are other regions in the world, in the US, where people are tested regularly to see if they're carrying it. And if they're in the healthcare professions, then they might not work with certain very susceptible populations. They might just be switched to a different floor or different ward at that point in time. Our next bacterium is in what we call the Nasseria genus, and its specific name is Nasseria gonorrhea. It is a gram-negative diplococci, and it is found pretty much exclusively in the human urogenital tract. So that really is the only place it can survive and that it can live. It's not carried by other types of animals. It's not carried in the environment. It's actually, when it gets in the body, it's pretty good at causing disease, but when it's outside of the body, it's a pretty weak bacterium. It's not good at surviving in the environment, fairly delicate. So as soon as it exits the body, it has a very, very short lifespan, and so it's not like something in the environment can harbor this and really transmit it to another person in most situations. There are some exceptions, but in most situations. In terms of virulence factors, one of the things that makes it so good in the human body is its fimbriae. The fimbriae are pretty extensive, they're very sticky, and it makes the bacteria be able to stick very well in the urogenital tract, even though that region of the body tends to be flushed fairly frequently with things like urine and other types of discharge. It also produces a protease, which breaks apart antibodies. So even when our body produces things like IgA or other types of antibodies to try to combat this particular bacterium, this bacterium is able to create this enzyme, this protease, that will break apart the antibodies that we create. So our antibodies are oftentimes not very effective at trying to combat this because of that. This bacterium is transmitted almost exclusively through sexual contact, but it can also be transmitted from mother to infant during the birthing process. So most cases of transmission are in a situation where there's been sexual contact between two or more people and the bacteria spreads that way. The other more common way for this to be transmitted is if a mother is infected with this bacterium while she's giving birth, the bacteria will oftentimes get into the infant's eyes and cause a fairly serious eye infection that needs to be treated if it happens. There are ways to help prevent this from happening during the birthing process. One obviously is testing just to make sure the mother is not infected with that. And then when a baby is born, they can put certain drops in the eyes to make sure that even if she was infected and either wasn't tested or the test was incorrect or inconclusive, the drops get put in there and help prevent any type of infection if the baby did come in contact with it. The infection that this causes is called gonorrhea. So just like its name says, this is the bacterium that causes the STI, the sexually transmitted infection that we call gonorrhea. Most infections of gonorrhea are limited to the genital region. There are exceptions to that. One of them we just talked about in the eyes of infants. but most of the time gonorrhea stays in the human urogenital tract and just kind of lives there. It is possible for that bacteria to spread throughout the body. If someone has an infection that has not been treated for a very long period of time, it's possible, although not necessarily probable, that this will spread from the reproductive tract to the rest of the body and start to cause problems there. So it's possible that someone can get what's called an extragenital infection. Sometimes those occur because of a genital infection that has spread. Other times they occur because it was passed on to a baby and it got into the eyes during the birthing process. And then unfortunately there is one other way to spread gonorrhea to a different part of the body besides the genital regions or the reproductive tract, and that is through what's called autoinoculation. Autoinoculation is a type of extragenital gonorrhea infection where someone who has a genital infection of gonorrhea touches the reproductive region or the genital region, gets the bacteria on their hand, and then doesn't clean their hand before they do something like touch their eye, their nose, or their mouth, and then they spread the bacteria to that part of their body as well as a result of moving it from the genital region to typically the head region, somewhere like that, because we have at least 3 places up there that can get infected easily: the eyes, the nose, and then the mouth. So good news about Nasseria gonorrhea is that it's typically fairly easily preventable as long as safe sex is practiced. And if someone does have this infection and they are going to give birth, they get either antibiotic treatment or those drops are put in the baby's eyes. Or again, if someone has this infection and they make sure to wash their hands after touching the genital region before they touch anywhere else on their body. Our next organism is also in the Nasseria genus, and its species is Meningitidis. So this bacterium is called Nasseria meningitidis, and it is also a gram-negative diplococci that is found pretty much exclusively in humans. This one, instead of being in the genital tract, though, is in the respiratory tract. And many people can be, and sometimes are, asymptomatic carriers of this particular bacterium. If you look at kind of any general population in the US at least, and this probably extends worldwide, but at least in the US for sure where the research has been done, between 3 and 30% of the people in that population will be carrying this bacteria in their respiratory tract without having any symptoms, meaning that the bacteria is not causing disease, it's not causing them to run a fever, it's not causing them to be sick or anything. They're just carrying it with them in their body. And for most people, the carrier state or carrier status lasts from a few days to a few months, and then they clear it from their body without ever knowing or without ever having any problems from it. In terms of virulence factors, this is another one that once it's outside the body, it's pretty weak and easy to get rid of, but when it's inside the body, it's got some pretty good virulence factors that help it survive pretty well and cause disease. It also has fimbriae that are very good at sticking to the respiratory tract, which is another region of the body that's actually flushed fairly frequently. So these bacteria are very good at sticking in there, despite the fact that there are fluids and there's air that's constantly moving through that region and moving things around. It also has the ability to produce a capsule, and there are 12 different types of this capsule. So person A in a population could be carrying this type of bacteria with capsule type A. Person B could be carrying this type of bacteria with capsule type B. Person C could be carrying it with capsule type C. Person D could be carrying it with a different types of capsule type. Basically, these different variations in capsules give the bacteria a little bit more of an advantage in terms of causing disease, because that means that we would need to produce different types of antibodies for each type of capsule to help fight it off. Now, in terms of the capsule types that are most likely to cause disease, like it says here, capsule types A, B, and C are the ones that worry us the most, because the bacteria that have those capsule types are the ones that are the most virulent in terms of being able to cause disease. It does also produce a protease, which will break down antibodies that we produce to it. And remember, in addition to just being able to break down the antibodies, because there are so many different capsule types, that complicates things for us even further and makes it even more difficult for our immune system to deal with this bacteria. In this case, it also uses LPS, which is called endotoxin, as a serious virulence factor. So if you think way back to Unit 1, you remember lipopolysaccharide, and that is a compound found in all gram-negative bacteria. And so all gram-negative bacteria have LPS, and another word for LPS is called endotoxin. But in this case, the LPS is particularly problematic for us because when this bacteria moves from the respiratory tract to the cerebrospinal tract, the LPS causes a very big reaction and our body causes a lot of inflammation, which ends up putting a lot of pressure on our brain, which is problematic. So LPS is dangerous all the time, but it, in this case, is especially problematic because this bacteria sometimes migrates from kind of the respiratory tract region to the brain, to the spinal cord, and that inflammation that it causes is very problematic in that area. In terms of the infection that this bacteria causes, we call the infection that it causes meningitis. Keep in mind that many things can cause meningitis. The term meningitis just means swelling of the meninges. And the meninges are tissue coverings that surround our brain and our spinal cord. So many bacteria and many viruses or fungi, etc., technically can cause meningitis. It's just in this case, we're talking about this bacteria that causes a case of meningitis. In terms of transmission, it is typically through direct contact or droplets. So either people touching each other skin to skin, or someone sneezing or coughing and spreading it that way. And most of the time, we see this bacteria cause problems in places where people are kind of crowded together. Things like dorms or barracks or other situations where people are living in very close contact with each other and spreading it around in kind of a very limited environment. In terms of treatment and prevention, when someone is diagnosed with this particular bacterial infection, they are given an antibiotic called a cephalosporin, which is a type of beta-lactam antibiotic. And they're given that particular type of antibiotic because those are able to cross what is called the blood-brain barrier and actually make it to the region of the body where this bacteria is and is causing the infection. Not all antibiotics can do that, but these particular ones can. For prevention, there is actually a vaccine, and that vaccine works primarily against types A, B, and C, although I think they've been able to kind of expand that recently and make the vaccine work against some other capsule types as well. And oftentimes this is given to not necessarily everyone, but people who are most likely to come in contact with something like this. Military recruits going off to basic training, college students who are going to be living in the dorms, and other regions again, or other situations again, where people will be living in kind of crowded environments, because that is typically where we see this type of infection occur. And then finally, another important thing to note about this one is when someone does come down with meningitis from this particular bacteria, the people that they have been around within the last couple of days or maybe the last week or so are given tetracycline antibiotics to help prevent them from getting the infection. So those antibiotics are there to help make sure the people who have come in contact with this person who has gotten sick don't also develop an infection. At this point, we have finished up our cocci and we're moving on to rods. Our first rod is one that's called Carinibacterium diphtheriae. It is a gram-positive rod that is found primarily associated with the human respiratory tract. We can find it in some other regions as well, but most of the time we find it pretty much exclusively in the human respiratory tract. And the thing about this bacteria is that it is not actually very harmful in and of itself. However, if it acquires the ability to produce a toxin as a result of a bacteriophage infecting it, then it becomes dangerous. So this bacteria itself is not a problem. It can just be part of the normal bacteria that's carried in human bodies as it always is. And it's not a problem until it gets infected with a bacteriophage and starts producing something called diphtheriatoxin. So that it says phage encoded, which just means the bacteriophage contains the genes for that toxin. And when the bacteriophage infects the bacteria, it gives the bacteria the ability to create this toxin. In terms of the infections that this bacteria can cause, once it has been infected with the bacteriophage, it can cause a local infection, a toxemic infection, or a cutaneous infection. In a local infection, what ends up happening is bacteria that are carried in the throat start to produce this toxin, and as a result, our body responds and produces what's called a pseudomembrane. So this is a really thick, sticky substance that is membrane-like in nature. It's not a true membrane, which is why it is called a pseudomembrane, but it's like a membrane. And if that infection is not noticed and not treated, eventually that pseudomembrane will grow across the entire throat and will cut off the air supply, which obviously is problematic, and it will lead to death. In terms of the toxemic infection, what ends up happening is the bacteria grow in the body, they produce this toxin, and for whatever reason, instead of having that pseudomembrane start to being produced in the throat, the toxins end up getting into the bloodstream typically. They start circulating throughout the body and start to damage the heart and the nerves by stopping protein synthesis in those regions. The least worst case scenario with this type of infection is a cutaneous infection, where typically the skin around the mouth will become infected and you will get that pseudomembrane kind of starting to form around the mouth. And certainly it can be uncomfortable and problematic, but most of the time this type of infection with this bacteria is not lethal, and you have a little bit more time to treat it before it becomes really problematic. In terms of transmission, this bacteria is primarily transmitted through droplets, direct contact, fomites, and these days contaminated milk. This was not quite as common a couple of decades ago, but now that we're starting to see the raw milk movement really gain traction and take place, we are starting to see some more people come in contact with it and get sick as a result. In terms of prevention, there's a great vaccine for it. Most of the time this vaccine is given in childhood, and it prevents a lot of childhood deaths and illnesses. And just kind of a fun fact about diphtheria vaccines and the treatment of diphtheria in general is that horses were actually kind of instrumental in helping create what was called the antitoxin for bacterial infections before there was widespread antibiotic use and before there was widespread vaccination. So when people got sick from diphtheria, obviously this was over 100 years ago, and it couldn't be prevented or couldn't be treated, they were able to often isolate the antitoxin from horses and use that to treat someone who was sick from this type of infection before they had other treatment means. Our next group of bacteria are those that are found in what we call the Clostridium genus. There are kind of four main ones that we will talk about, and we'll get into those in just a second. But just some general characteristics about this group of organisms. They are gram-positive spore-forming rods, and they are found in many environments. So it is very common to find any number of different types of Clostridia in the soil, in water, and sometimes they're even carried typically in low numbers by many different types of people. So many people might have them on their skin in low numbers, in their intestine in low numbers, and then they're just found generally kind of everywhere in the environment. In terms of the most significant species to humans, at least, we have Clostridium perfringens, Clostridium botulinum, Clostridium tetani, and Clostridium difficile. So starting with Clostridium perfringens, this organism causes a condition we call gas gangrene, or sometimes just gangrene. And the way it does that is that it produces various toxins and enzymes that start to degrade cells and degrade tissue. Additionally, it often goes through the process of fermentation once it has infected the body. And as it goes through fermentation, it releases gas, and that gas starts to build up in the tissue and cause the area that's been infected to become puffy. In terms of where people pick this up, usually this is one where if someone gets some kind of wound, especially when they're out in the environment, and they get some kind of soil in it, or perhaps the organism is in the water and their wound gets exposed to the water. If the wound is not cleaned properly and is not looked after properly, this infection may start to develop. For treatment, there are antibiotics that we can use that can help deal with the infection a little bit, but really the big one for this is oftentimes when someone has this type of infection, they go to sessions in what's called a hyperbaric oxygen chamber. This is a chamber that has very high pressures of oxygen, and while that person is in the chamber, that oxygen is basically forced into their body, either in through their lungs and just into the bloodstream, or directly into the wound. And by creating more oxygen in the body, then this organism actually gets poisoned. So this is an obligate anaerobe, and the extra oxygen helps to poison that organism, kill it off, and that allows the body to start to heal. Our next one in the Clostridia is Clostridium botulinum. This organism causes what we call botulism, and most of the time when people come in contact with this organism and the toxins that it produces, it is through improperly prepared food. And when I say improperly prepared, most of the time I'm talking about improperly canned food. So the canning process is a pretty specific process, and when it is not done properly, then some of the organisms from the environment can get into the can, and when that can is sealed up, the organism starts to grow. It releases a toxin called botulinum toxin, which is thought to be one of the most potent toxins in the world, and that ends up causing uncontrollable muscle relaxation. And while being relaxed may sound nice, it is not great when we're talking about the muscles that are involved in your lungs or muscles that are involved in your cardiovascular system. Those ones need to contract periodically to make sure that we are getting oxygen and other types of things flowing throughout our body. This is another interesting one because if you have ever heard that you are not supposed to give infants under 1 year old honey, this is the reason why. So like I said, most of the time when people come in contact with Clostridium botulinum, it has to do with improperly canned food, but it is possible to pick this up environmentally. And when you are an adult, or at least you're older than about 1 year old, it's typically not a problem if a few of these are in your food, because your normal bacteria that live in your intestine kind of take up all that space and they make it so that this organism can't cause any problems. However, with infants under 1 year of age, they don't have quite as many bacteria living in their intestine. And so if they are exposed to this organism in their food, then it is possible they will develop what we call infant botulism as a result, and the organism will grow in their intestines, will release this toxin, and will lead to what's sometimes called floppy baby syndrome. So it is very common to find spores of this organism in honey, and that is why they tell you not to give honey to infants under a year old. Our next one is Clostridium tetani. This one primarily causes what we call tetanus, and it enters the body most of the time through wounds. One thing that's important to keep in mind is that it's not just through things like rusty nails. A lot of times people assume that you can only get tetanus from something rusty. That's not true. You can absolutely get tetanus from something that looked completely pristine. You can get tetanus from a rusty nail. You can get tetanus in a lot of different scenarios. So it's not just something rusty that can give you tetanus. It is possible for the spores of this organism, or this organism in general, just to enter through the umbilical cord as well if it is cut with an unsterile instrument. So that doesn't happen very often in the US or other developed nations, but in certain situations, especially in developing nations, if someone gives birth in a hospital or a clinic or at home and the umbilical cord is cut with something that's not sterile, it's possible to get that organism into the infant through the umbilical cord. Now with this one produces a toxin that almost does the exact opposite of the botulinum toxin. This organism produces what's called tetanospasmin, and it causes uncontrollable muscle contractions. So that's what you're looking at down here. This is kind of a classic example of the types of muscle contractions that we might see in tetanus. And it is fatal in some cases, especially if you're talking about younger kids or infants or the elderly. Many, healthier people can survive this, although it's, from what I hear, incredibly painful. I have never had tetanus, fortunately. And one of the interesting things about this organism is that tetanospasmin starts to work first, most of the time, on the jaw. So the muscles that open and close the jaw, that's where the toxin tends to work first. And as a result, another term for this disease or this condition is lockjaw. So if you hear someone having lockjaw, it means they have tetanus. And basically what's happened is they've gotten this organism in their body. The organism has started to produce this toxin. The toxin has caused the muscles around the jaw to contract, and the person can't open them, which is certainly problematic. And then our last organism in this genus that we'll talk about is called Clostridium difficile. Clostridium difficile is one that many of us will carry with us in low numbers in our intestine, and typically it's not causing a problem. However, occasionally it can start to overgrow, start to produce some toxins that cause intestinal damage, and lead to lots of bloody and yellow diarrhea that can be fatal in certain circumstances. So oftentimes this one will start to become a problem when someone has been either in the hospital and has acquired more than normal of these spores in their body, or if they've been treated with long-term antibiotics that have destroyed the normal flora that live in the intestine. In either or both of those cases, what can happen is the bacteria starts to grow in the intestine, produces these toxins that cause a lot of intestinal damage and lead to very large quantities of diarrhea that can be both bloody and yellow in color, and also, from what I've told, has a very, very distinct smell to it. So it's something to be careful of. These spores happen to be very easy to pass on from patient to patient. So sometimes we find a situation where one patient on a floor will come down with what's called C. diff, and then many other patients on that same floor will come down with it as well, just because it's very, very easy to transmit the spores from room to room as healthcare personnel go from room to room to check on their patients. We are moving on from our gram-positive spore-forming rods and talking about a couple of bacteria called mycobacteria. So mycobacteria are what we call acid-fast rods. Now these are technically gram-positive, meaning they have a relatively thick layer of peptidoglycan that covers a cell membrane. However, they also have mycolic acids, which are types of lipids that are very sticky and very waxy, and they kind of cover this layer of peptidoglycan. So these rods or these organisms, while they, like I said, are technically gram-positive, the gram stain doesn't work on them because reagents like crystal violet and the iodine, they can't penetrate or they can't make it through these mycolic acids. So we do have to use a special stain to dye these organisms. We use carbofuscin and we do the acid-fast procedure, which you guys have done in lab at this point, I imagine. And the big thing about these organisms is that they don't have a bunch of virulence factors except for these mycolic acids. So these mycolic acids are a pretty big deal because they really help protect the organism from things like phagocytosis and from getting degraded by antibodies or being worked on by antibodies. They protect them from things like complement and basically all of the defenses that our body has against bacteria don't work as well on the acid-fast bacteria because of these mycolic acids. There are lots of different types of mycobacteria, but the two most significant for humanity are Mycobacterium tuberculosis and Mycobacterium leprae. Mycobacterium tuberculosis causes the disease that we call tuberculosis. It is found almost exclusively in humans. We really don't find this particular organism outside of humans, or at least associated with anything but humans. So this one can live for a significant amount of time out in the environment, but if we find this organism, it tells us that a human has been there at some point. So it's not like this one's really carried by animals very often or anything like that. In terms of the infections that it causes, like I said, tuberculosis, and there are different types of tuberculosis. There is primary tuberculosis, which primarily means that the bacteria are infecting the lungs, and you can see over here on the upper right-hand side, this lung infection here, kind of spread out white, kind of spider web-like or cobweb-like infection here. And if this is treated appropriately, most people can get over a case of tuberculosis. It does typically require a long course of antibiotics, and oftentimes it requires multiple different types of antibiotics. But if it's treated properly and caught soon enough, many people can get over this infection. They can clear the organism from their body and they'll be okay. There are some cases, though, where either the person wasn't treated soon enough, they weren't treated at all, or the antibiotics didn't work, and they will develop what's called latent tuberculosis. With latent tuberculosis, essentially what happens is the bacteria are just hanging out in the lungs. They may be at very low numbers, but they aren't gone. So these bacteria just wait there until conditions are good. They come out of latency and they cause a second instance of disease or a second course of tuberculosis. Oftentimes the second course is a lot worse than the first one. And this is definitely one where if it is not treated, the prognosis or the outlook is not very good for that person, because by this point the bacteria has had a long time to spend in the body. It may have started to spread to other regions besides the lungs, and it's starting to really do some damage to the tissue. And then finally, there's also what we call extrapulmonary tuberculosis. This oftentimes happens after a latent tuberculosis infection kind of flares up again. Not always, but often. But extrapulmonary just means that the bacteria have spread throughout the body and they've moved away from the lungs and they started to infect other regions. As long as this is caught soon enough and taken care of soon enough, again, this is a situation where the person probably has a decent chance of survival. But again, it may have done a decent amount of damage to the body as well. And so even if they survive it, there may be some long-lasting damage to the other organs that it has infected. In terms of transmission, tuberculosis is primarily transmitted through droplets. The interesting thing about these type of bacteria is they can oftentimes survive up to about 8 months or so outside the human body. Most of them aren't going to survive that full 8 months, but there are individual bacteria that can seem to survive up to 8 months outside the human body and remain infectious. So this is one that it tends to take a lot of bacteria to make someone sick and have a lot of exposure to it. But once they do get sick, it tends to grow fairly well in the lungs because, again, those mycolic acids really protect it from things like phagocytosis. They protect it from destruction. It protects it from dehydration when it's outside the body. And so really those mycolic acids play a significant role in causing disease because it just makes it so that our defenses aren't very good against it. Now, in terms of our next organism, Mycobacterium leprae, this causes what we call leprosy. And this is a disease, actually tuberculosis has been around a long time too, but leprosy is definitely a disease that has been causing problems for humanity for thousands of years. We find this organism associated with humans and armadillos, strangely enough. So for a long time, there was a question about whether did armadillos give it to us or did we give it to armadillos? And the most recent research suggests that we gave it to armadillos. So armadillos, if you know what an armadillo looks like, it almost looks like this little opossum that has kind of armored plates around it. And if you flip that armadillo upside down and underneath those kind of armored plates, it will have little fuzzy hairs. Oftentimes, those armadillos will carry this bacterium in those little hairs in that area of their body. In terms of the infection it causes, like I said, leprosy. Oh, it's written down there. You didn't have to copy it from up there. But anyway, it causes leprosy, which is also called Hansen's disease, because there was a physician/scientist whose last name was Hansen who did a lot of work with this. And what this organism does is, just like with Mycobacterium tuberculosis, it doesn't have a ton of virulence factors except for those mycolic acids. And so those mycolic acids allow it to survive in the body, resist phagocytosis, and just keep multiplying slowly over time. What it ends up doing is causing tissue damage, especially in the extremities, towards the hands, the ends of the fingers, and the feet, the toes. And oftentimes, this particular bacteria doesn't cause any problems aside from the fact that it is there and doing damage that way. But because it is there, sometimes we lose sensation in our fingers and in our feet. And so we might get some kind of cut or some other kind of wound that we don't really recognize. And what ends up happening is we get a secondary infection that causes more problems. And we end up with situations where we have to either amputate part of the extremity, or it just ends up kind of dying and falling off naturally in certain circumstances. In terms of treatment and transmission, the transmission actually still isn't fully known. This is a bacteria that's very difficult to work with in a laboratory setting. It's very difficult to model, and it's very difficult to understand the transmission of it. And in terms of treatment, it's another one that's difficult to treat because a lot of times there aren't a lot of obvious symptoms for it. Initially, some people may get kind of a skin rash in certain areas, but sometimes that'll go away, or they don't really think anything of it, or there's nothing really they can do about it anyway, and so the disease continues to progress. If it's caught early enough, usually antibiotics will work against the bacterium. Again, oftentimes it's a long course of antibiotics, and maybe multiple antibiotics will be involved, but they do usually work. The issue is, though, that oftentimes we don't catch it soon enough because there aren't a lot of obvious signs and symptoms for it. So by the time we notice something's wrong, the disease has progressed and it has caused significant tissue damage already. Moving on from our acid-fast rods, we are going to move into gram-negative rods, and our first gram-negative rod is Pseudomonas aeruginosa. Pseudomonas is an organism that we find basically all over the place in the environment. We find it in all kinds of places in the soil and in the water. It is also carried by about 10% of any given population in their GI tract. So this is one that, probably because it is found in the environment so often and we're exposed to it fairly frequently, many of us will carry it in our GI tract kind of on a regular basis. And this organism is almost like the gram-negative rod version of staph. It's not quite as bad as staph, but it has incredible metabolic diversity. It's able to produce a bunch of different toxins and enzymes, and it's really able to infect a variety of places in the body. So Pseudomonas is another one of these that is a pretty nasty bacterium once it does start to cause infection. Most of the time, it doesn't cause a problem for us, but when it does, it can cause some really difficult to deal with infections. In terms of specific virulence factors, it is oftentimes very resistant to disinfection. Things like soaps, dyes, antibiotics, and other types of standard disinfection procedures don't always work on it. There are instances where you can actually find Pseudomonas growing in or living on soap. So especially bar soaps. Pseudomonas is just very metabolically diverse. It's very sturdy out in the environment, and it is very good at living in conditions that would kill most other bacteria. It produces a wide variety of toxins, a wide variety of enzymes. Both of these can do a bunch of damage to our tissues in various places in the body. It has a pretty, again, sturdy slime layer. That slime layer is very good at protecting it from our immune system. It's very good at allowing it to stick both environmentally and in the body. It is very good at using all kinds of different things for electron donors and electron acceptors. Basically, it means it can get its nutritional needs met and its ATP production needs met in all kinds of places in the human body. It also has a lot of LPS, so as all gram-negative organisms do, lipopolysaccharide is a significant virulence factor. In this case, its LPS is just very good at stimulating our immune system, and in the process of stimulating our immune system, sometimes our body does more damage to our tissue than the organism itself does. In terms of some of the specific places Pseudomonas can infect, you are looking at the lungs. Those are very common, especially in people who are immunocompromised or have a condition called cystic fibrosis. It can cause systemic infections. Either the bacteria gets into the blood or the lymph or the cardiovascular system, which would get it transmitted to the blood, and it would spread around the body, or its toxins or enzymes get spread around the body. It can cause skin infections, it can cause urinary tract infections, it can cause ear infections, it can cause eye infections. This is especially common with contacts. And one of, I guess I would say, the fun things about Pseudomonas is that many strains will produce this green pigment. And so not always, but at least sometimes when someone has an infection, something like the skin or the eye, we can tell it's very obviously Pseudomonas because it has a green tinge to it. Either the pus will be kind of green tinged, or their eye will take on a green tinge. And I've never really smelled it. It doesn't smell like this to me, but some people say that Pseudomonas smells like grapes. So that's interesting. In terms of transmission, it is transmitted in lots of different ways. It can be transmitted directly from person to person, so close contact, skin to skin, or something like that. It can be indirect transmission, so someone could cough and spread it and leave the droplets of that somewhere in the air or on a surface. It could just be through something like a pen or pencil, something like that. Fomites are very common in the transmission of Pseudomonas, things like sponges and washcloths, medical instruments. All of these can harbor Pseudomonas, and remember, Pseudomonas is very good at resisting disinfection. So even if you've taken standard disinfection procedures, they might not always work on Pseudomonas. We also find Pseudomonas in lots of different types of bodies of water, and that can lead to infections as well, especially in compromised people. In terms of prevention, most of the time when we know someone has a Pseudomonas infection, ideally there will be some kind of extra sanitization or extra sterilization procedures, or additional disinfectants or antiseptics will be used to help reduce transmission of that, or help reduce the ability of someone to give that infection to someone else. There is a vaccine under development. It's not ready yet, and it's probably not one that would be given to everyone, but if it gets ready in the next, hopefully, 10 years or so, then it might be one that we give to people who are more, are at more risk, who are defined as high-risk individuals for developing Pseudomonas infections. So people who are generally healthy, who have good immune systems, strong immune systems, they probably wouldn't get this vaccine. But for people who may be immunocompromised or have some other type of situation going on that would put them at more risk for developing a Pseudomonas infection, they would probably be eligible for this vaccine if it gets finished. Our next gram-negative rod is Bordetella pertussis. This is found primarily associated with humans. We can find it in other types of primate species as well, but most of the time we're talking about finding this associated with humans. In terms of virulence factors, it has a very good capsule that helps protect it from the immune system, and it produces a toxin that comes in two parts. So this toxin is a fairly large protein molecule, and it has kind of part A and part B. And one part of this toxin binds the cilia in the respiratory tract. So I'm going to see if I can zoom in without distorting things too much. Not great, but here you go. Trying to keep it even. Okay. So when we look at this bacterium, it produces this toxin, like I said, that's part A and part B. And one part of the toxin will end up binding the cilia. So the cilia are projections that come out of certain cells in our body, especially in the respiratory tract, that are used to help kind of flush and make things flow in the respiratory tract, so that anything that gets in there can be flushed out or can be gotten out, and it doesn't make it too deep in our body. But this bacteria is very tricky because it produces this toxin that will basically paralyze these cilia. This toxin will kind of fit in between those cilia and make it so they can't move anymore. Then, once those cilia are stuck, they can't move anymore, the second part of the toxin will enter the cells and cause those cells to produce additional mucus. So now this cell is producing additional mucus, but it can't use its cilia as normal to help clear that mucus. And essentially what happens is when someone gets this infection, they develop excess mucus in their respiratory tract they can't clear out. And if it is not treated properly, they basically just drown in their own mucus. This infection that is caused by this bacterium is called pertussis, or whooping cough. It's transmitted between people almost exclusively by droplets. And this is a disease where most of the time adolescents and adults, unless they're very elderly and weakened or weakened, can usually fight off this infection pretty well. So if an adolescent or a relatively healthy adult gets this bacterial infection, they will probably be okay. However, this is very, very dangerous for babies, for young children, for the elderly, and again, potentially anyone who is immunocompromised, especially if they already have issues with their lungs. So there is a fantastic vaccine for this particular bacterium. Actually, it's not against the bacterium itself, it's against just the toxin that is produced. But what we found recently is with the anti-vaccination movements, more and more people are choosing not to get this vaccine. So we're starting to see a rise in the number of cases of babies, infants, and sometimes even adolescents with whooping cough because of the lack of vaccinations. This is also one where the protection is finite. Basically, the vaccine works to get people through the most dangerous part of their life, which is infancy and young childhood, and then the protection starts to wear off. So by the time someone's a teenager or a young adult, most of the protection they had from the vaccines that they might have had as an infant has worn off, and they are again susceptible to this disease and also able to acquire this bacterium and start spreading it to other people as well. So if you are recommended to have a booster shot for pertussis, even when you are an older adolescent, a young adult, or even an older adult, the reason for that is, one, to protect you and to get your protection back up, but two, to protect younger people around you, especially if you're around younger children a lot, you're around infants. It's to try to make sure that you are not transmitting this organism to them and spreading this disease to a person that could be very vulnerable to its effects. We are coming up on our last 2 bacteria for this class. Hopefully you have enjoyed things so far. Again, like I said, we can't cover every single bacterium in here. I wish we could, but we cannot. However, I've tried to give the highlights of some of the most common bacterial infections that we deal with in the US and in some cases worldwide. So we've moved through our cocci, our rods, and we are going to move on to our spiral-shaped bacteria. Our first spiral bacteria is in what we call the genus Treponema, and specifically we are talking about Treponema pallidum. This is a gram-negative spirochete. In terms of the distribution, it is an obligate parasite of humans, which means this is another one that we really can't find outside of the human body. This is one almost kind of like Nasseria gonorrhea that once it exits the human body, it is very weak. It is very easy to disinfect for. It is very easy to get rid of, and it does not survive much longer probably than an hour max outside the human body. In terms of virulence factors, this organism has a very low infectious dose. We actually know exactly what number of organisms it takes to cause disease in a person is, and that is 57. It has a hooked tip that allows the organism to grab onto our tissue and kind of bind in and burrow in, almost the way a corkscrew would work. This organism stimulates the immune response, and interestingly, it has very few membrane proteins that our body recognizes as foreign. So it does have some antigens on its surface. It has some proteins that our body recognizes as foreign proteins. And so we are able to respond to it. However, it doesn't have a lot of them, and that makes it more difficult for our body to kind of trace this organism as it moves through our body and also to make antibodies. Remember, antibodies are made in response to antigens, and antigens are primarily the proteins that we find on the surface of microbes. There are different types as well, but most of the time when we're talking about antigens, especially in a microbiology context, we're talking about the proteins that are on the surface of a microbe. This microbe doesn't have a lot of proteins that our body can recognize as foreign. It has a few, but not a lot. And so that makes it that much harder for our body to trace it as it moves through the body and also produce antibodies that would actually work to kind of stop it from causing disease. In terms of the infection that it causes what we call syphilis. And syphilis itself has 3 stages if it is untreated. In the primary stage of syphilis, which lasts basically from the time that someone becomes infected until maybe a week, 2 weeks after the time of infection, a person will develop a sore. Usually it's kind of an open sore. It doesn't have to be very large, but it's called a chancre. There's different pronunciations of it though, so you may hear it in different ways. I'm going to write it out here. So some people call it a chancre, some people call it a canker. Don't mix that up with what a standard canker sore is. They're not the same thing at all. Some people call it a chancre. It really just depends kind of on where you are in the world and also to a certain degree where you are in the US in terms of what pronunciation is used. Nevertheless, however it is pronounced, a person who is infected with this organism will develop this kind of small, usually kind of small, open sore in the region of infection. Usually that is in the genital region, and oftentimes it's something that people don't really pay attention to because the sore itself actually heals. As the organisms start to move deeper into the body, they move away from that initial site of infection. So the sore heals up and it goes away, and people just don't really think anything of it. They're like, oh, okay, I had like a little sore there. Maybe it was an ingrown hair. Maybe it was just my clothes rubbed in a certain way. Whatever, it's gone, I'm better. Unfortunately, in this case, they are not better, and what that is actually a sign of is the bacteria moving deeper into the body. In this situation, the bacteria starts to get deeper into the body, and most of the time, people will develop a very light rash, either on their abdomen or sometimes on the palm of their hands, or maybe even the soles of their feet. And again, this is one that many people may not even recognize, they might not see it, or they might not really think anything of it. So if they don't get treatment at this stage, the bacteria continues to move into the body, and they enter what's called the tertiary stage of the disease. The tertiary stage is one that can oftentimes kind of go unnoticed for a very long period of time. What's happening in the tertiary stage is the bacteria is migrating to areas of the body, like the brain and the cardiovascular system. And it's starting to infect the very, very deep parts of our body. It may take 20 years or so for someone to notice that they have tertiary syphilis, but once they notice it, typically the bacteria has done fairly extensive damage to the brain, to the spinal cord, and to the cardiovascular system. And although they could still potentially take antibiotics and get rid of that infection, the tissue damage will have been done, and a lot of times it can't be healed. Now, most of the time, at least in the US, people do not develop tertiary syphilis because they either get treatment during primary syphilis or at some point during the time where they have secondary syphilis, even if they don't know it, they get some other kind of infection that they get antibiotics for. Maybe they get strep throat, or they get bronchitis, or they get some kind of skin infection. And so they take antibiotics to treat this other obvious infection, and those antibiotics happen to work to get rid of the syphilis as well. However, in developing nations where people may not be getting antibiotics, it is easier for them to progress from primary to secondary to tertiary without getting treatment, because this is one that sometimes it's not obvious you are infected with. Not everyone will recognize the symptoms of it, and in some cases, even if they do recognize they have symptoms, they don't really have any options anyway because there isn't a healthcare provider nearby, or the prescription is too expensive, or there just isn't the infrastructure to get them the medications. So this is a disease that, if it is caught early enough and treated early enough, most people will be okay. However, it's one of these situations where if it is not caught, this can do serious damage to the body that will last the rest of that person's life. Now, most of the time, this is acquired as a sexually transmitted infection. However, it is possible for this bacteria to be passed from a mother to a developing fetus. Not many organisms can actually cross from a mother's body, make it to the placenta and through the placenta, and then make it to the developing fetus, to the baby. However, this is one that can. This bacteria can make it through the placenta and infect a developing fetus and cause long-term damage to that individual. So this is one that most of the time pregnant women are screened for, just to make sure that they are not carrying it, that they are not infected, because this is one of the few that can cross that placenta, make it to the baby, and start to cause some problems. Like I said, in terms of transmission, we really basically went over that. Primarily, it's through sexual contact. It can be from mother to fetus. And in terms of treatment, our general antibiotics still work pretty well as of now. We may start seeing some resistance in the next 10 years or so, but hopefully we'll develop some new antibiotics and we'll not see a lot of resistance overall. Hopefully we can kind of keep up with this one and make sure that people are not moving into this tertiary stage of the disease. We have made it to our last bacteria that we'll talk about, and that is Helicobacter pylori. This is a gram-negative curved rod, so technically it's a vibrio. It looks oftentimes like a spiral-shaped bacteria, and some people do call it a spiral shape, but technically from an anatomical perspective, it really is a vibrio rather than a spirochete or a spirillum. Nevertheless, the more important aspects about it are that most of the time we find this bacteria associated with the human GI tract. In terms of virulence factors, primarily the two things it does is that it produces just mechanical or physical damage in our body by burrowing into tissue. And it does also release some enzymes as it does that kind of further degrade the cells in the area. Now, for a long time, it was thought that this organism was just part of our normal bacteria flora, no problems, didn't do anything to us. However, there were some scientists who started realizing that patients with stomach ulcers tended to have this bacteria associated with them. Now, at the time, the thought about ulcers was that people who are type A personality, who drink a lot of coffee, who are under a lot of stress, those are the ones that get stomach ulcers, and no one else really does. These ulcers are caused by stress or by lifestyle, and people just need to change those things and they'll be better. However, these scientists/physicians realized that that might not be true. They had a lot of patients who maybe either didn't fit into this type A personality category, or who maybe they were type A, but they had this bacteria, and this bacteria kept kind of coming up. It was this common theme in these stomach ulcers. So these scientists proposed that maybe this bacteria was actually the cause of stomach ulcers. And no one really believed them until they did an experiment. And I will stop the lecture right here and just remind you, this is not something to try at home at all, ever. But these scientists/physicians decided to test their hypothesis that this bacteria was actually the cause of stomach ulcers, at least in 90% of the cases. So what they did was they decided to take images of their GI tract, and as expected, there were no problems, there were no ulcers, everything looked normal. Then they drank solutions of this bacteria. So they grew this bacteria in a lab, put it into a culture tube, and they drank that culture tube. They waited a little bit, and they developed ulcers. They did imaging of those ulcers, took those pictures. Okay, they have the ulcers now. Then they treated themselves with antibiotics. Did the imaging again, the ulcers were healed. So that alone was not necessarily enough to show definitively that those bacteria were the cause of the ulcers, but additional research was done after that, And it was shown later that about 90% of stomach ulcers are caused by this bacterium. And so now, by showing that most of these ulcers are actually caused by a bacterial infection as opposed to some kind of lifestyle situation, we have a way to more effectively treat these conditions that people will develop. The researchers who did this also ended up winning the Nobel Prize, but again, just a reminder, this is not something to try at home. So in addition to causing stomach ulcers, this bacteria can be associated with a certain type of cancer called adenocarcinoma or adenocarcinoma. The reason for that is not that the bacteria necessarily does something to cause the cancer exactly. It's just that repeated infections or an infection that can't get cleared up by antibiotics or can't get under control by antibiotics leads to this tissue damage, and as the body tries to repair itself, there's a chance that mutations will occur during the process of mitosis and cellular regrowth, and because of that, then the person will develop cancer as a result of this infection, essentially. So it's not that the bacteria directly causes cancer, it's just that the bacteria causes damage, and in certain cases, while the body's trying to repair that damage, mutations occur in the cells, and that leads to cancer developing in certain regions of the body. In terms of transmission, it is primarily transmitted through either the oral route or the fecal oral route. And unfortunately, we have found that at least some transmission seems to occur as a result of houseflies. So houseflies will either, this is a really bad situation, but land on fecal matter, and they'll land on something someone eats or drinks, and they will obtain that bacteria that way. Or there will be a situation where the housefly will land on something someone is eating or drinking, pick up the bacteria that way, then go land on something else that someone else is eating or drinking and drop off the bacteria in that situation. In terms of treatment, antibiotics work to clear up most cases of these very well. Antacids are often given as well to help kind of protect the area where the ulcer is. So the antibiotics help deal with the bacterial infection, and then the antacids will help protect this damaged area while it heals, hopefully. So that is our last bacterium. Next, we will move on to viruses, and then we will finish up talking about things like protozoa, helminths, fungi, etc.